Hi, I'm Lisa Miner. I'm from Multispan, and I'm going to talk to you about how working with Multispan, we can enable drug discovery with you with cell engineering, assay development, and high throughput screening. So the things I'm going to talk about in my presentation are, who are we? Who is Multispan? What do we have? So what cell line offerings do we have for high throughput screening and lead optimization? I'll tell you a bit about our beta-arrestin technology, which we introduced recently last year. And then I'm going to talk to you about new offerings that we have, including additions to our beta-arrestin technology and some other stuff. And then in addition, some case studies showing what it's like to work with Multispan. So here is a history of Multispan. Multispan, we were incorporated in 2004. We were WSOSB certified in 2019 and MBE certified in 2020. Our expertise is cell engineering, assay development, and high throughput screening. We're a United States-based company. One of our biggest assets is engineering and cloning stable cell line for targets. We have included now CRISPR knockout to that technology. We do assay development using both primary and recombinant cells, high throughput screening, lead optimization screening, and compound profiling. Our biggest expertise is GPCR, and we're considered best in class for GPCR targets. But we also have expertise in other things, including kinase, transporters, nuclear hormone receptors, just to name a few. We have patented multi-screen beta-arrestin sensor technology, which I'll talk about, and GPCR expression technologies as well. We have been told that we have uncompromising high quality and integrity with how we work with clients and our general way we work with people. We have 100% client retention year over year, which speaks to the quality and the integrity. This is basically our functional and clonal stable cell lines for high throughput screening and lead optimization. We have over 500 well-validated stable cell lines. We started out in 2008 with only 104, and over the years we have expanded to, like I said, over 500. This is mostly GPCRs, but it does include receptor tyrosine kinases, zinc transporters, and integrins, potassium channels, etc. On the right-hand side of the slide here is our functional GPCR safety assessment panel. This safety assessment panel was developed not just by us, but essentially by partnership with pharma companies on what kind of functional assays do they need for safety assessment. And so we've included ones that were basically high interest that folks would want. And this is available on demand anytime somebody asks for it. This is an example of our high throughput screening assay platform using the mu opioid receptor. One of the advantages of our cell assay technology is that we've set it up so essentially you can do multiple assays in the same cell. So that actually eliminates the comparison between two cell types or two cell lines when evaluating a signal. So this is an example looking at cyclic AMP, looking at receptor internalization. We also can do that. Looking at beta-arrestin beta-arrestin recruitment, GTP gamma S binding, saturation binding, and ERK phosphorylation. And again, this is all in one cell, so it makes it easy to be able to interpret the results, especially when you're looking at signaling bias, like for cyclic AMP versus beta-arrestin. We also have assay-ready cells. So this is basically cells on demand. These are frozen cells which can't be propagated. They're cheaper because you can only use them once. It saves time and cost and compound screening. You don't have to do any assay development. You don't have to do any cell culture with them. Essentially, we sell them to you along with pre-validated assay kit, like for cyclic A, calcium, beta-arrestin. When you get this, essentially you can run your assay and throw away the cells and you're done. The advantage of this is that if you don't have a big need for a lot of assays, or you just want to spot check something, or you don't have a lot of money, this essentially is one way to to basically make this work. And on the right-hand side, this shows the serotonin receptor family and all the different things we have. So these are all the different receptors, the different species, stable cell lines. We have division-arrested cells, which is what I'm talking, the assay-ready cells, membranes, and then the different assays as well. We have a beta-arrestin technology. Our beta-arrestin technology is based on essentially complementation. And in this complementation technology, we have an arrestin, which is tagged with the luciferase, half a luciferase essentially, and the complementary half a luciferase is in the membrane. When the receptor is activated and the arrestin translocates to the receptor, there is binding between the complementary parts with luciferase, and then you get a signal. This actually is a really nice assay in that the GPCR is not tagged at all. And so you can use it on native GPCRs, you can use it on anything that's untagged, and that works very well. This complements and rounds out our GPCR technologies. So we have receptor G proteins, essentially GTP gamma S, cyclic A, calcium, IP1. We also have GRK2, beta arrestin or phosphorylation internalization, as well as the internalization in the other assay platforms. So the benefits of this is that there's no GRK2 co-transfection required. It's a really easy protocol, and it can be done in basically using a stable cell line or transfected into primary cells. So this is looking at beta arrestin in CHO and HEC293 parental stable cell lines for untagged GPCRs. So essentially we have here the HEC293 cells and the CHO K1. These are parental cell lines with the arrestin that's parental there all the time, and you transfect your GPCR into that. So here this looks at the beta arrestin with mu opioid receptor with a HEC293, and then the CHO cell line as well. This is a beta arrestin and cyclic A assay for 4 members of the proton sensing family. So this is with the GPR4, 65, 68, and 132. Looking at calcium, we can do calcium, cyclic A, and beta arrestin. And here again is your beta arrestin assay with the stable cell line. And this is the cyclic A assay as well. So essentially you can look at but two assays in one cell. So beta arrestin and cyclic A. So in 2020, we have added some things. So we've added the arrestin sensor technology for native untagged GPCRs using a delivery system, either BACMAM or HSV virus. We've also added CRISPR stable cell line generation. We have high throughput screening now. So we've increased our screening capability essentially by adding some tools to do that. We can do 100,000 compounds with a 1-month turnaround and lead optimization with a 1-day turnaround. We've also dipped our toe into the COVID-19 world, and we're able to we have stable cell lines with RNA-dependent polymerase of CoV-2, and then 3CL protease as well, and then the ACE2 stable cell lines. So you can actually do cell-based assays for that. I'll talk just a couple seconds about that, but really that's not the focus of this discussion. So these are assay-ready reagents to measure beta arrestin recruitment. So we have the advantage of this is we have it develops a high titer arrestin sensor viral particles. The assay is ready really fast in 24 hours. We have efficient delivery into both primary and recombinant cells. And this is an example looking at the beta arrestin BACMAM delivery and then HSV delivery with either the GLP-1 or the kappa opioid receptor. And you see very nice signals with the beta arrestin recruitment. We're also able to do target to lead with our help, helping our clients to be able to build and screen their assays. So one of the advantages of working with Multispan is we can develop the assay or use one of our cell lines that we have. When we do the assay for you, we do the assay without charging for the cell line, without charging for any of the reagents. We just basically charge for the screening. So we have a fast turnaround, 1 day for lead optimization, 1 month for 100,000 library with high throughput screening. We have free access, like I said, to all of our cell lines and reagents. Here's an example of mu-opioid receptor with beta arrestin. Here's an example with the CHOK1 with the G-alpha-16. This is our signal and how good the Z prime is with this. So we're able to do with very consistent delivery our assay. The other thing is when we do these assays for people, we agree, we always run it with a QC. We agree with the client what the QC is going to be. And if the assay does not pass the QC, we rerun it at our cost. So it's always make sure that you have quality data that comes with our results. So here's an example with the GLP-1 high throughput screening assays. This is just with human here with the cyclic A, arrestin. Again, this is within the same cell. But we also in this have some cynomolgus monkey, rat, dog, and mouse in the primary signaling assay. And that way you can look to see and make sure that your assay works for the orthologs that you're going to test your compounds in. We also have developed CRISPR technology for knockout. Here is a beta-2 gene-edited clones. When we do the CRISPR technology, we do a functional assay to assess whether there's what the functional loss of activity is. And here is a complete homozygous knockout is in blue. The heterozygous knockout here is in green, and the wild type is in red. And this is with the beta-2. We can do COVID-19 drug discovery. Remember, I said that we have developed stable cell lines for COVID-19, and I'm not going to talk about that, but you can look in our website and actually see more details about what we've been doing and what partnerships we've made with this. So now I'm going to talk a little bit about some case studies, because it's one of the things about working with us, not just knowing our reagents and our work and stuff, but how do we work with people? So this is our case study looking at compound profiling to support a fixed panel of GPCRs. So in this project, we actually had a farmer reach out to us for a trial study. And again, this was a competition with 2 other providers. And the idea was to provide a small set of lead compounds and a family of 10 inflammation-related GPCR targets. And this was a competition, like I said before. And with this competition, we successfully delivered and won a long-term contract. The phase 2 of this is ongoing. This is a blanket PO, 6 to 10 screens per year, 1 week turnaround for each cycle. Our data has been published by the customer. This phase 2 has been renewed annually, and it's still going strong after 5 years. So our stable cell lines were delivered, and we actually were able to deliver this contract and win the contract, and still are working with the clients. Case study 2 was a virtual drug discovery nuclear hormone receptor. This was a Bay Area VC. They were interested in a nuclear receptor. And so this is actually something this is for oncology. They needed high throughput screening. They didn't have anything. So we provided guidance on the screening funnel, including primary and secondary assays. We custom developed the suite of assays and to support the screening campaign. We worked with a CRO collaborator to provide biweekly lead optimization support. So at this point, the lead candidate is in preparation for IND. This has been a 2-year project. We support the programs basically from scratch to IND by doing all the in vitro screens that are necessary. So the compounds from our work, just to let you know, have successfully entered the market. And although we do GPCRs, we do other things as well. And this was a nuclear hormone receptor project. So this is stable cell lines for partial agonist screening. In this is a large pharma. We had been working with this company for over a year. for over 10 years, sorry, to provide hand-in-hand stable cell line and assay development support. This company approached us for advice to help them identify partial agonist cell-based assay development for screening program. The assay exploration was in transient cell transfection. We used tool compounds to look at the results to determine the correlation between expression level and pharmacology. The phase 2 was a stable cell line clone development for the human target. With our clients, we chose and validated using the full and partial agonist tool compounds. And again, one of the things about working with us is that we are in constant contact with our partners and discussing with them the results, going over the data, choosing the right clones for the projects. And that's one of the things we try to be as transparent as possible with all of the results that we have. So phase 3 of this was comparable stable cell line clones and assay development with 6 orthologs targets using the human target pharmacology as a benchmark. So we delivered 7 stable cell line clones with assay protocols on time. And we've worked with this company for a long time and keep supporting the same team for years. And case 4, again, is another competition. This was a biotech pharma looking for existing cell lines and assays. They wanted 2 stable cell lines expressing a human target, high and low, validated in calcium and cyclic A. They wanted 2 stable cell lines expressing the ortholog, validated in cyclic A. So the first thing was a competition with 2 other providers. The scope was to provide tool compounds in the human target and multiple assays, profile tool compounds in the human target, multiple assays. And once again, we were able to outcompete the competition and won the contract. So phase 2 is lead optimization support under a blanket PO. This is weekly primary screen with next day data delivery. We can do this because we have everything set up so that we receive the compounds and the assays run the same day. So next day data delivery. Secondary screen is ortholog and binding assays and then signaling bias. So essentially we're able to fill the gap in screening funnel in parallel, doing custom cell line assay development for human target radioligand, beta-arrestin, and canine target assay. And this is still ongoing. This is phase 2A. It's renewed annually, and this has been 2 years, and it continues. So just wanted to remind you that we've helped our abilities to be able to provide cell lines, assays, high-throughput screening support, lead optimization support, have helped companies be able to move compounds into the clinic. And that for us is really a primary importance. For primary assay development, our key considerations we always consider is what signaling pathway, what splice variant, what expression levels, and we want to make sure the parental is good. Secondary assay development is also important, looking at orthologs and off targets, et cetera. And then the SAR campaign, the turnaround time is critical. So in summary, multi-span capabilities, we consistently demonstrate that we can engineer and clone quality, reliable, stable cell lines. We develop quality assays using recombinant and native cells for GPCRs, but we also do kinases and other targets, and I showed you a nuclear receptor as an example. Our GPCR assay portfolio is very large and includes multiple assay formats and multi-screen arrest and sensor technology. We have unparalleled quality turnaround time for high quality data, 100,000 compounds in high-throughput screen in a month, routine SAR, primary assay in a day. Like I said before, all assays have to pass QC as agreed to us by us and the client, and if they don't pass the QC, it's repeated at our cost. 7 of the top 10 pharmas are active customers, and we have 100% repeat customer rate. And thank you for your attention.